Good afternoon, everyone. I am Arthon Greenspan, a sophomore at UC Santa Cruz.
And I'm William Chang, a freshman attending Northwestern University.
And we are presenting automated analysis of astrocyte cell connectivity after laser ablation using machine learning and pathfinding algorithms.
Before we get into all the technicalities of our project, we'd like to acknowledge our mentors and our collaborators. First, we have Dr. Gomez, who was our assistant project scientist, and Dr. Linda Shi. Dr. Linda Shi, our PI, actually spearheads a program, a summer internship at UCSD called OPALS, the Outreach Program for Advancing Learning in STEM, which is where Connor, Arthon, and I met. Now let's get into breaking down this long project title. We'd like you to get a basic understanding of what astrocytes are and laser ablation is before we get into our methodology. So first of all, a question for the audience. What do you think of when I mention the cells of the brain? Any answers? Yes?
Neurons?
Neurons, that's right. So neurons are the potent electrochemical communicators of the brain. But if you've ever noticed anything about neurons, you probably know that they're long and thin and very easy to damage. So what does the central nervous system do about this? It has to fix this fragility issue some way. Well, we have astrocytes. And astrocytes are known as the unsung heroes of the nervous system because they serve a variety of functions, from structural support to being the miniature immune system of our brain tissue. Before we advance on to the next part of our methodology talk, I'd also like to help all of you keep in mind that astrocytes are important when it comes to understanding neurodegenerative diseases and brain trauma, because we look at the responses of these astrocytes to understand how brain tissue as a whole responds to such abrasions. And how exactly do we induce damage in the lab, you may ask? Laser ablation is the answer. In our lab, the Biophotonics Center at UC San Diego, we have a high-powered MiTi femtosecond laser that is directed through a complex mirror setup and can basically induce damage in a straight line in any type of cell. As seen here, this is a collaboration between our lab and the University of Queensland in Australia, where they etched out the names of both institutions on a couple of red blood cells.
And with that, we arrive at the problem. This is the data that we have to process. And now I'm a robotics engineer. So when I have to figure out what is the, calcium intensity over time and the connections between the cells, I don't know anything about biology. How am I supposed to figure that? Well, we take a human-in-the-loop approach. We need an intimate connection with the data and how people look at this data and how they solve these problems. So I got to meet with Dr. Gomez, and she walked us through how this is done manually, typically in a lab. As a biomedical engineer, like, how is this done? What data do I look for? And through this, I got 3 key takeaways that I wouldn't have gotten otherwise. So, first of all, the nuclei all on their own is not enough to get normalized calcium intensity. If you look back at these images, notice how all of these nuclei are almost the same color. That's because the robolase hasn't actually gone into the nucleus. So you need the nucleus and the surrounding cytoplasm to get normalized calcium intensity. Second of all, this is a video. This is one image of a 500 frame video. And so which is the best frame to do all of our analysis on? Well, it turns out there is one optimal answer, and that is the frame before laser ablation. Because if you go too early, the robolase hasn't had time to sink in yet, and if you wait too long, then the laser ablation makes it really hard to figure out what is happening to the cells. So the frame before laser ablation is the best choice. And finally, the microscope that we are using only takes images that are 4 micrometers thick. And this is going to be really important for the future because that means that we are working with a very 2D problem. There is a small 3D aspect, but it is not significant, and that's going to be very important for our final implementation. So now we get into our algorithms of choice. First one, cell pose, which is a segmentation machine learning model that has a really interesting like teacher-student relationship where you actually, it makes a guess and then you manually correct and supervise it, and then it takes the next image, and you're sort of tutoring it until it's ready to go. Really cool read, check out this paper if you have the time. And second, one of my favorites as a robotics engineer, A-star pathfinding. So let's explain how all of this goes together. We start with our image, and we use two cell pose models, one trained for nuclei, one trained for cytoplasm. We can then take the nuclei and assign them to a cytoplasm. Multiple nuclei can go to the same cytoplasm. If they don't find a cytoplasm, no problem. We assign it one based on the statistics of the image. No big deal, we've got a backup. With this, we can calculate the correct normalized calcium intensity and get our graph parts of the image. Part one done. We've done it. All right, but we still need to network these cells together. We need to figure out how they're related. And we could use another machine learning model for that, but because of the knowledge we have of these images and the nature of the data set, we don't need to. We can use A-star pathfinding by assigning the background, which is anything more than 1.21 standard deviations below the mean, to a wall, and everything else as a constant cost path. We can just check, is there a path? If there is, they're networked. If they're not connected. And because of the nature of how thin our image is, this is a reliable technique. And so we can use machine learning when it is needed, but when it is not needed, we can use algorithms to complete our graphs. And so we go from a video file to 500 images to getting our relationships and our data, and we finally automatically output our graphs.
And in addition to our graphs, once we receive all of the data in the form of calcium intensity, we're able to categorize all of these different cells. Are they networked? Are they not?
Yeah.
And from that, we understand how cells interact and respond to trauma.
And so now, we'll be able to get to our conclusions.
Wait, hold up. We have to first talk about some of the experiences beyond just the technical skills.
Absolutely. Beyond being able to work on an incredible project, we learned a variety of soft skills as well, such as how to communicate with industry professionals, how to take notes, how to ask good questions, and also just learning more about what our futures could potentially be.
And moving to my first year as a OPALS intern, I learned that as someone interested in biology, biology and computer science go hand in hand. As you can see here, and in this picture, manual quantification and analysis is incredibly time-consuming and inefficient. There's also user bias when it comes to analysis of the pictures, which these algorithms hand in hand can help to resolve. We also would like to note that these are the same cells, but using a different microscopy technique. And returning to the OPALS program as a mentor this past summer, I noticed a lot of commonalities among all of the different OPALS projects, and that is, there is so much that could be resolved with the potential use of the integration of pathfinding and machine learning algorithms that has not yet. I'm a biology student. I work in a biology lab in college. In immunology, the same thing holds true. So much manual quantification is needed, and yet we haven't found a solution to accelerate that research using computer science, and we think we have the solution here now. And it doesn't just extend to the other OPALS projects that we've provided our interns. It's also a variety of different fields, from brain trauma, which is our current focus in this project, to the broader field of computational neuroscience. Now, in neuroscience, you have all of these neural networks. You have these nervous tissues communicating with one another. So it's especially important that you find the connections and you see how those connections play into response to injury. And finally, neuropharmacology. That last picture I showed you on the last slide, that is actually interns looking at how cells are responding to a type of drug, CBX, that is proven to slow down the rate of neurodegeneration. And with that, we'd like you to leave with knowing that in the field of science and in a variety of different fields, there is a lot of potential for this integration. Thank you so much for listening, and feel free to reach out to us if you have any
Emails.
Any questions? So, can you model this as a graph theoretical problem also? Like it's a pathfinding problem?
Oh yeah, I think A-Star does model it as a graph. We just left, like, it's pretty simple for us because we just need to figure out if there is a path,
I
not
see.
the best path.
I see.
And so this is a graph, but okay, so every node that is below the background intensity is a wall, and then all of these just have the same cost.
Oh, I see.
For a
So-
truck. They all have a cost of 1, yes.
So is this like a 2D slice?
Yeah, it's a 2D slice.
Yes.
So would you like also pathfind on like third dimensions?
We don't have to, because it's a 2D slice. So we're taking advantage of the fact that it is a 2D image to be able to use this algorithm.
And I'll just add on very quickly, in most biology labs, even the labs looking at more complete tissue samples, the maximum depth of any sample is about 20 microns, and that's still incredibly small. You can treat it as a 2D image.
So, and for our images, there's a maximum of 2 layers possible, and so we can solve for the first layer by just directly drawing on it, and then the second one only has one case where there's a cell under the 2, but that can be solved by just realizing that there's 2 sides sticking out of it, and because of the nature of how an astrocyte is shaped, the only possible solution is that they're connected, which is also solved by having a cell on top of it. So this works for both of those cases, yeah. So we're taking advantage of the fact that, like, the main thing we're like arguing for here is that you get a lot of benefits by, like, instead, like, there was an alternate universe where I just had 2 terabytes of data thrown at me with no context. And so what we're arguing for here is you get a lot better results when you go and talk to the people, because it tends to be that the fields who need these problems solved and the people that have the ability to solve the problems are 2 completely different fields. And so you need to, like, you get a lot better results when you cross-communicate and you learn the process. So we actually sat down and, like, watched someone manually do all of this to get to this algorithm.
That is that's what we were advocating for.
Yes.
In being able to
Human in the loop.
connect,
Yes.
connect different fields and have the human in the loop.
Yeah.
Thank you, guys. So, yeah, I think
We're good? Yeah.
thanks.
Yeah.
So,